All right, today we're decoding the Krebs cycle. This is the central metabolic engine that powers nearly every single breath you take and every move you make. So let's dive in. So you know how glycolysis breaks glucose down into pyruvate? Well, it turns out our cells have only just started to tap into its energy. The Krebs cycle, which you might also hear called the citric acid cycle, that's where the real energy extraction begins. Think of it like a metabolic furnace. It systematically burns or oxidizes carbon molecules to generate this huge payload of high-energy electron carriers.
Okay, so before the main event can kick off, we've got to get our star player, our key molecule, to the right place and, in the right uniform.
And that right place? For eukaryotic cells, like the ones that make up you and me, it's deep inside the mitochondria, in what's called the mitochondrial matrix. So the pyruvate that was made out in the cytosol has to get transported into this inner chamber of the mitochondria before anything else can happen.
This is where a super crucial link reaction happens. It's called pyruvate oxidation, and it's all about prepping the fuel for the cycle. So here's what goes down. First, that 3-carbon pyruvate molecule gets into the matrix. Second, one of its carbons gets chopped off and released as carbon dioxide. Yep, the same stuff we breathe out. Third, the 2-carbon piece that's left over hooks up with something called coenzyme A, and boom, we get our key molecule, Acetyl-CoA. And as a bonus, this whole process spits out our first high-energy electron carrier, a molecule of NADH.
All right, acetyl-CoA is ready to go. Now we can finally enter the cycle itself. Let's walk through one complete turn, step by step.
So the interesting thing is this 2-carbon acetyl-CoA molecule doesn't just kickstart the cycle, it joins it. It finds a 4-carbon molecule called oxaloacetate that's just hanging out, waiting. They combine, and what do you get? A 6-carbon molecule called citrate. And hey, that's exactly why the Krebs cycle is also called the citric acid cycle. Make sense, right?
Okay, from this point on, it's basically a series of 8 enzymatic reactions. You can think of it like a molecular relay race. Citrate gets passed from one enzyme to the next, getting converted and changed at each step, and releasing energy and carbons as it goes. But here's the really cool part, the thing that makes it a true cycle. The very last step regenerates that 4-carbon oxaloacetate we started with. So it's a self-perpetuating loop, always ready for the next acetyl-CoA to jump in and start the race all over again.
So, what's the big prize for finishing this race? Well, for every single turn of the cycle, we get a few key things. First, it spins off 2 molecules of carbon dioxide as waste. It also makes 1 molecule of GTP. Think of it as a close cousin to ATP, just another small packet of energy. But here's the main event, the most important part. It loads up our high-energy electron carriers. We get 3 molecules of NADH and 1 molecule of FADH2. These guys are the real treasure.
Okay, time to do a little math. Let's figure out how much energy we're actually getting out of that one single glucose molecule we started with. Now you gotta remember, glycolysis splits 1 glucose molecule into 2 pyruvate molecules, right? So that means for every single glucose, the Krebs cycle has to run twice. It's a 2-for-1 deal. So if we just double the output from a single turn, the grand total from 1 glucose molecule is pretty impressive. 6 NADH, 2 FADH2, and 2 GTP.
But what's the whole point of making all these carrier molecules? Let's zoom out and look at the bigger picture to see why the Krebs cycle is just so incredibly important.
And this, right here, gets to the absolute heart of the cycle's true purpose. I mean, the direct energy you get from that little bit of GTP is tiny. The real goal of the Krebs cycle is to act like a harvester, collecting all these high-energy electrons and loading them onto our carrier molecules, the NADH and FADH2. They're like little energy taxis.
So now, these fully loaded energy taxis, our NADH and FADH2, travel to the inner mitochondrial membrane, and here they drop off their precious cargo. They donate their electrons to the electron transport chain. This is a series of protein complexes that uses the energy from those electrons to generate the vast majority of the cell's ATP. We're talking somewhere around 34 times more. This is the big energy payoff we've been waiting for.
Okay, so this pathway is obviously fundamental to life. But how do modern scientists, in this age of genomics and big data, actually study and analyze something this intricate?
Well, that is exactly where bioinformatics comes into play. Researchers use these incredibly powerful databases, like KEGG, That stands for the Kyoto Encyclopedia of Genes and Genomes. And what it is basically a massive collection of detailed maps for pretty much every known metabolic pathway.
And when you look at these maps, you see that the Krebs cycle isn't some isolated little loop. No way. It's a central hub, a major intersection connected to hundreds of other things happening in the cell. And by using bioinformatics tools, scientists can actually analyze how the flow of molecules through this pathway changes. This allows them to directly link malfunctions in the cycle to diseases like cancer and metabolic disorders. It's become a super critical focus in systems biology and, of course, in drug discovery.
So to boil it all down, if you remember one thing about the Krebs cycle, remember this simple flow. Acetyl-CoA goes in, it turns the cycle to pump a bunch of electron carriers out, and those carriers are used to generate a ton of ATP later. In, out, payoff.
So there you have it. The Krebs cycle really is the tireless central engine of cellular respiration. If you want to decode more complex topics in biology and bioinformatics, make sure you subscribe to BioInfo Bytes. Thanks so much for watching.